Welcome to Membrane Structure and Function, Part 1. You'll remember from NYA that the cell is surrounded by the plasma membrane. This membrane not only encloses a cell, but surrounds many specialized organelles and divides it into compartments. The plasma membrane and the various internal membranes of cells consist of a double layer of phospholipids with proteins attached to or embedded in it. The plasma membrane functions as a selective barrier that allows sufficient passage of oxygen, nutrients, and waste to service the entire volume of the cell. Membranes are vital because they separate the cell from the outside world. They also separate compartments inside the cell to protect important processes and events. They are of crucial importance to life because a cell must separate itself from the outside environment for two major reasons. It must keep its molecules of life, DNA, RNA, and the assortment of proteins from dissipating away, and it also must keep out foreign molecules that damage or destroy the cell's components and molecules. However, while the cell must abide by these two principles, it must also communicate with the environment to continuously monitor the external conditions and adapt to them. For example, if an E. coli bacterium detects a high concentration of lactose in the surrounding environment, it begins synthesizing proteins to take in and metabolize the lactose. But if the E. coli also detects a high concentration of glucose in the environment, it begins instead to synthesize the proteins to take in and metabolize the glucose. It also needs to pump in nutrients, release toxic products of its metabolism. But how does an E. coli know? It depends on its membrane proteins to gather information about the environment in various ways. Cells within complex tissue of a multicellular organism also need to exchange metabolites and waste products with the outside, but they require information that is far more complex than that demanded by bacteria. Each of the cells in our tissues communicates with dozens, if not hundreds, of other cell types about a variety of issues: when it should grow, if it should differentiate or die, when it should release certain protein products or metabolites needed by other cells at distant sites in the body, and what other cells it should associate with in order to build the complex tissues. Ironically, the intrinsic structure of a cell creates a formidable obstacle to these important processes of intercellular communication. The shell shields itself behind its membrane, which is relatively impermeable to most types of molecules. This barrier enables the cell to create its own intracellular environment that is distinct and very different from the world around it. But it also creates a problem in that it prevents the free exchange of materials between the inside and the outside. It must be breached somehow for a cell to inform itself on what is happening in the outside world. And again, you'll remember from NYA that phospholipids are amphipathic, which means that it has a hydrophilic or polar head, which is glycerol, phosphate, and organic base, and two nonpolar hydrophobic tails, which are fatty acids. This particular group of phospholipids, called the phosphatidylcholines, have a choline group attached to the phosphate group, and in fact are a major component of cell membranes. Because phospholipids are amphipathic, when they are added to water, they will self-assemble into aggregates known as micelles that protect their hydrophobic portions from the water. These are very important for the transport of hydrophobic compounds in the blood. Phospholipids will also form bilayers. This is the reason that they are the major component of a cell's membranes, both internal and external. One of the very earliest episodes in the evolution of life may in fact have been the formation of a membrane that enclosed a solution different from the surrounding solution while still permitting the uptake of nutrients and the elimination of waste products. The capacity of the cell to discriminate in its chemical exchanges with the environment is fundamental to life, and the plasma membrane molecules that compose this make it selectively possible. Thus the plasma membrane not only forms a boundary between the cell and its surrounding, it in fact controls the traffic of molecules in and out of the cell. It permits the uptake of nutrients and the elimination of waste products. The plasma membrane is selectively permeable. That is, some substances can cross easily, others have a more difficult time, while others cannot cross without help. Because the cell surface provides the only opportunity for interaction with the environment, since all substances enter and exit by the plasma membrane, it plays a key role in controlling cell function. Once again, we will be looking at how form fits function, and one of the most important features of biological membranes, the ability to regulate transport across cellular boundaries. We're looking here at a muscle cell, which has a steady stream of small molecules and ions that move across the plasma membrane in both directions. Consider the chemical exchanges here. Sugars, red, amino acids, and other nutrients enter the cell, and metabolic waste products leave the cell. The cell takes in oxygen for cellular respiration and expels carbon dioxide, so we could see the waste perhaps as green. It is important to note that the cell also regulates the ion concentration in organic ions, such as calcium, potassium, sodium, and chloride, and the cell is able to shuttle them one way or the other across the membrane. Despite the fact there is extensive traffic through the membrane, they are selectively permeable, and you don't have indiscriminate movement of substances. The cell functions as a barrier, a semi-permeable barrier. The cell can take up certain small molecules and ions and excludes other. And in fact, the movement that does occur occurs at different rates for different substances. And as we can see, there's a high concentration of sodium outside the cell, whereas there's a high concentration of chloride and potassium inside the cell. The selective permeability of the membrane makes this possible. And here we see a model of a membrane. Here we see the relative permeability of a synthetic lipid bilayer to different classes of molecules. Here we see that very small nonpolar molecules can simply diffuse across the membrane, and that even if there are uncharged polar molecules, if they're small enough, like glycerol and water, they too can diffuse across. Large uncharged but polar molecules, such as glucose, cannot cross the membrane, and ions are completely excluded from crossing the membrane. A typical membrane found in cell organelles contains about 50% protein by mass, and the remainder is mostly lipids. The relative amounts of lipids and proteins vary among different membranes. Some membranes, such as the inner mitochondrial membrane, have no carbohydrate with their proteins, while the plasma membrane of a eukaryotic cell can have a very large amount. Despite the fact that it's 50% protein by mass and the remainder is lipids, the lipid molecules are much smaller and outnumber the proteins by about 50 to 1. Half of a phospholipid bilayer is called a leaflet. Each leaflet faces a different region. For example, the plasma membrane contains a cytosolic leaflet facing the cytoplasm and an extracellular leaflet facing the cell's exterior. With regard to lipid composition, the two leaflets of the cellular membranes are highly asymmetrical. The most striking asymmetry occurs with glycolipids with attached carbohydrates. These are found primarily in the extracellular leaflet of the plasma membrane, so that the carbohydrate portion of a glycolipid protrudes into the extracellular medium. In 1917, Irving Langmuir made artificial membranes by adding phospholipids dissolved in benzene to water. Following the evaporation of the benzene, the phospholipid remained as a film covering the surface of the water, with only the hydrophilic heads of the phospholipids immersed in water. Not much later, two Dutch scientists reasoned that the cell membrane must actually be phospholipid bilayers, 2 molecules thick, because it could exist as a stable boundary between two aqueous compartments. So if the phospholipid bilayer is the main fabric of a membrane, where do we put the proteins? Despite the fact that the heads of phospholipids are hydrophilic, the surface of an artificial membrane consisting of a phospholipid bilayer adheres less strongly to water compared to a real biological membrane. And this could be accounted for if the membrane were coated on both sides with hydrophilic proteins. The Danieli-Davison model studied these triglyceride phosphobilayers over a water surface, and they produced a model which in fact had it as almost like a sandwich, a phospholipid bilayer between 2 layers of protein. Today we know that proteins float around in the membrane, forming multiple coils that weave in and out of the membrane, with tails of the proteins projecting out on both the outside and the inside of the cell. And this would be the Singer-Nicolson model. This model would be called the fluid mosaic model. In this model, the membrane is a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. The fluid mosaic model is continuously being refined. For example, groups of proteins are often found associating in long-lasting specialized patches where they carry out common functions. The lipids themselves appear to form defined regions as well. Also, the membrane may be much more packed with proteins than imagined in the classic fluid mosaic model. Though membranes are often described as fluid, it's more appropriate to say membranes are semi-fluid. In a fluid substance, molecules can move in 3 dimensions. By comparison, most lipids can move freely only in 2 dimensions, around their long axes and laterally within the membrane leaflet, because rotational and lateral movements keep the fatty acid tails within the hydrophobic interior. Such movements are energetically favorable. In fact, at 37 degrees, a typical lipid molecule exchanges places with its neighbors about 10 to the 7th times per second and can be directional, moving several micrometers per second. At this rate, a lipid could traverse the length of a bacterial cell, which is approximately 1 micron, in only 1 second, and the length of a typical eukaryotic cell in 10 to 20 seconds. In contrast to rotational and lateral movements, movement in the 3rd dimension, the flip-flop movement of lipids from one leaflet to the opposite leaflet, does not occur spontaneously. Energetically, such movements are extremely unfavorable because the polar head of a phospholipid would have to be transported through the hydrophobic interior. Because movement in the 3rd dimension is limited, the 2 leaflets of a membrane phospholipid bilayer can be distinct from each other in phospholipid composition. This difference relates to biological functions that occur on each surface of the leaflet. The biochemicals of sterols and phospholipids have a profound effect on the fluidity of the phospholipid bilayer. 3 major factors play important roles in membrane fluidity: the length of the fatty acid tails, the presence of double bonds in the acyl tails, and sterols that are interspersed throughout the membrane. So we're looking at membrane fluidity. The fatty acid tails of the phospholipids range in length from 14 to 24 carbon atoms, with 18 to 20 the most common. Shorter tails lead to fewer stabilizing interactions, and therefore a more fluid membrane, whereas longer tails lead to more hydrophobic interactions and more stabilizing. A second important factor is the presence of double bonds in the fatty acid tails. When there is a double bond between carbons, the lipid is said to be unsaturated with respect to the number of hydrogens that can bond to the carbon atoms. A double bond changes the shape of the fatty acid tail and prevents the close packing of hydrocarbon tails and reduces hydrophobic interactions, consequently making the bilayer more fluid. A third factor is the presence of sterols interspersed throughout the membrane, particularly the plasma membrane. In animals, the dominant sterol is cholesterol. Cholesterol tends to stabilize membranes, but its effect depends on temperature. At lower temperatures, such as in the icy water, cholesterol tends to act as a spacer between the fatty acid chains, preventing stabilizing interactions and thus maintaining fluidity. However, at higher temperatures, such as those observed in mammals that maintain constant body temperature, the single hydroxyl group of the cholesterol reacts with the hydrophilic head of the adjacent phospholipid, restricting its motion and making the membrane less fluid and preventing the membrane from becoming unstable. In most plants, cholesterol is not common, but other sterols, such as methylcholesterol, regulate plant membrane fluidity, and their relative amounts may help plants adapt to changes in ambient environment. Cells can adapt to changes in temperature by altering the lipid composition of their membranes. An optimal level of bilayer fluidity is essential for normal cell function, growth, and division. If a membrane is too fluid, which can occur at higher temperatures, it can become leaky. Conversely, if a membrane becomes too solid, which may occur at lower temperatures, the functioning of the membrane proteins will be inhibited. So, for example, when the water temperature drops, certain fish will incorporate more cholesterol in their membranes. If a plant cell is exposed to high temperatures for many hours or days, it alters the lipid composition to have longer fatty acid tails and fewer double bonds. Phospholipids and sterols can move laterally within their leaflet with ease, but there are certain membrane domains called lipid rafts that are distinctive regions of phospholipids enriched with sterols and membrane proteins. The lipids of a lipid raft tend to have fatty acid tails that are straight, that are hence saturated, and can pack more tightly together. They are thus more stable and tend to stay clustered as a unit, which is called a raft. The name lipid raft derives from the fact that the phospholipids, sterols, and proteins remain associated with each other and float together in the semifluid membrane. They are often regions in which specific membrane proteins are inserted, and as we'll see later, they are often involved in cell signaling. This is a Harvard University animation that you may have seen before, red blood cells and white blood cells moving along the inside of a vessel. Here we see adherents, and here we are looking at the plasma membrane from the outer side. So these are the hydrophilic heads of the phospholipids, and this is what we had talked about, a lipid raft. So these are made up of different phospholipids that pack very much more tightly together and move as a unit through the phospholipid bilayer. And they have very special proteins that are associated with it on the extracellular as well as on the intracellular side. We see the lipid raft moving. Here we see the lipid raft moving. Here we're on the cytosolic side. And the lipid raft, here we are underneath, we're inside the cell, and there's the lipid raft with a different protein that's associated. And as we'll see later, when we will be talking about cell signaling, proteins on the outside can act as a lock, and when the key fits in, that changes the shape of the lock, which then initiates a change in another protein in the lipid raft. And as I said, these are associated with cell signaling. So thank you for listening to Membrane Structure and Function Part 1. We'll be continuing in class on Tuesday, and then we'll be looking at membrane transport.